forage is not going to save the bees. And that's related to neonics and on some of these other pesticides. The neonics aren't staying put. They're getting into the water. They're getting into the soil. And they're getting into untreated plants. On one corn seed, there is enough insecticide to kill 360,000 bees. On one corn seed. Neonicotinoids are 5,000 to 7,000 times more toxic than DDT was. Sorry folks, GMOs are nothing compared to neonics. It's a drop in the bucket. We decided, this was one of my former postdocs, Dr. Chrissy Lundgren. We decided if we feed the bees a little bit better, if we give them good forage, maybe we can offset some of the adverse effects that the neonics are having on the bees. Just like us, maybe we can withstand certain stresses if we're well fed. So we planted these conservation mixes next to cornfields that were treated and then on organic cornfields that had never been treated. If you're ever looking at how to design a good conservation mix, the answer is simple, it's diversity. That's what you want. You want a lot of different flower colors, you want a lot of different flower shapes and sizes, and different flower architectures and plant architectures out there. You want diversity, okay? You want flowering periods that last the whole year. Is this the perfect blend? No. Is there such a thing? No. But this one worked pretty good, okay? And then we went out and we collected honeybees and nectar and pollen and honey from each of these sites, and we analyzed it for the amount of clothianidin, one of these neonics. And we also looked at the nutritional status of the bees. How healthy are they? Okay. Clothianidin. This is all in the leaf tissue. All of our conservation strip mixes were hot for the neonic in the leaves. 4 parts per billion is the LD50 for honeybees. That's the lethal dose for 50% of the bee population, is right there. 4 parts per billion. We find that there's learning disabilities at 1 part per billion. Reduced reproductive capacity of honeybees at 1 part per billion. Have you ever tried to get the nectar out of a flower using a fine glass capillary? It's like a little straw in the field. It's a royal pain in the ass. The wind is blowing. It's like, but the bees are really good at it, right? And so we let the bees, we collect the bees from the plants that they were foraging on, and then we rip out their honey stomachs, and we analyze the nectar that they had just consumed for some of the neonics. And it was all hot at a much lower rate, but it was still hot. Here's honey, here's pollen. All hot. Pollen the bees were collecting from the environment had 10 times the LD50. But what was most alarming about this study is that the organic farms had just as much insecticide as the conventional farms. Organic doesn't mean organic anymore, folks. Sorry. These compounds are not staying put. Clothianidin quantity in the bees. This is the protein, fats, and carbohydrate storage in the bees. Essentially, as the clothianidin goes up, we see nutritional stress on those bees going up. Or, yeah, the nutritional status goes down. So the bees, or the neonics, are certainly correlated with the stress that's on these hives. Never fear, right? Because the next generation of pesticides is here. Have you guys heard of RNA interference?
Oh, that sounds bad.
Yeah, so this is RNA interference is a new form of genetic pesticide that they're starting to, well, it was just commercialized this year. Essentially what they're now able to do is they can flip off genes through ingested nucleic acid called RNA, and they can flip off any gene that you might want to in a particular pest. And so we can kill pests by flipping off critical gene function in the pest. So this would be in line probably with some of the synthetic biology ideas and things along those lines, I think. So what they've done is they've genetically engineered corn. This is now commercially available to flip off key genes in the stomachs of corn rootworm beetles, which is one of the key pests of corn. And now what they're doing is they're developing RNAi-based insecticidal sprays. And because throughout much of the country, weeds have become resistant to glyphosate, Roundup. And so what they're going to do is they'll spray on RNA that will then flip off that resistance gene in the weeds so that they'll become susceptible to glyphosate again. So we did one of the first real exposés of sorts on risks of RNAi-based pesticide technology. This was back in 2013 when Monsanto was really getting hot to trot for this technology. And we pointed out that there are actually risks associated with it. These products are marketed as it is harnessing the power of the genome to be very, very specific for your target organism. And what we pointed out is that there are risks, namely that you accidentally flip off the wrong gene. So you could flip off the correct gene, like that stomach protein gene in the rootworm beetle, but in a different organism. Okay, so silencing the correct gene in the wrong critter, or you could accidentally silence a completely different gene in the wrong organism. So those are two very critical risks, right? And so we tried to, this is very complicated. Cable Hardin is an animator friend of mine who works at South Dakota State. And if you want to see this video that kind of explains what RNAi is in layman's terms, as well as some of the risks, it's just Google this and it should come up. And we're going to try to play that video now. Let's see what happens.
DNA, deoxyribonucleic acid. It's the genetic material that's in the cells of all living organisms. It's the double-stranded helix structure in cell nuclei that carry the genetic information of an organism. It consists of thousands of genes that are made up of millions and billions of pairs of nucleotides. Each gene is a set of instructions on how to build a protein molecule. But in order to make a protein from DNA, there's another player, messenger RNA. DNA is copied into mRNA, RNA, ribonucleic acid, a single-stranded sort of mirror image temporary version of DNA. This mRNA transfers the DNA message from the nucleus to the ribosomes, where the proteins are manufactured. The mRNA is then translated into proteins. And in turn, these proteins do things. They are the workhorses for cellular and organism function. Taken together, DNA, RNA, and proteins are the blueprints and raw materials for life. Knowing that these molecules exist in living organisms and how they work gives humans some powerful tools for manipulating the genetics of organisms. Well, this brings up another molecule involved. It's a version of RNA, that temporary copy of the DNA, called RNAi. The I in RNAi stands for interference. When a cell wants to stop making a protein, it produces a little RNAi molecule which silences certain DNA from producing a protein. RNAi is a naturally occurring, necessary genetic component of all organisms, including humans. For example, RNAi has the important job of fighting things like viruses and regulating genetic changes and mutations. RNAi does this by specifically targeting certain sequences of DNA and blocking the production of proteins. Since the discovery of the RNAi process in the 1990s, this genetic mechanism has led to some pretty innovative applications. Application of RNAi has shown to be a promising method of improving life for us on many levels, including switching off genes that cause diseases, learning what genes do and how they work, and making food production easier. Researchers use RNAi by designing and introducing short strands of RNA, around 21 to 25 nucleotides. These short strands bind to the complementary sequences in the genetic code. RNAi works by stopping the information in the DNA from getting to the protein-making ribosomes. It interferes with the messenger RNA. When it comes to agricultural applications, RNAi can be used as a form of genetic pesticide that can be built right into a plant's biology. For example, an insect pest feeds on a crop that deploys RNAi coded to stop the ability for that pest to digest food and process nutrients from the plant. As a result, the pest's growth or its ability to reproduce is slowed or halted, or the pest dies. Or a crop produces RNAi that changes a plant's chemistry, making that plant unattractive to a pest. Or the RNAi can block a plant's susceptibility to an herbicide, allowing the herbicide to only kill weeds on the farm. When we consider how society relies on crops for food, fuel, and fiber, it's easy to see why RNAi can be a valuable asset in crop improvement and a powerful tool against yield loss. However, just as when using other powerful tools, we have to make sure that the technology is safe. Keep in mind that genes are made up of millions and billions of pairs of nucleotides, and RNAi targets gene sequences around 21 to 25 nucleotides long. That's right, 21 nucleotides in a sequence out of billions in a gene. With those kinds of odds, the chances of the RNAi blocking other RNAs from producing totally unpredicted proteins is likely. One possible risk is that the RNAi molecule might silence the correct gene, but in the wrong organism. It turns out that the RNAi and the RNA don't have to be 100% identical for there to be silencing. For example, in addition to silencing part of a corn pest's digestive system, maybe an RNAi molecule would accidentally silence part of the digestive system of a lady beetle, or a honeybee, or a cow, or cousin Mabel. These risks are not trivial, especially because they're different from those posed by most other types of pesticides that we're used to dealing with. Now, considering the unintentional turning off of unintended genetic functions of targeted and non-targeted organisms, and the huge complexity of biological and ecological systems on which all life depends, this could be a bit of a problem. Without more knowledge about how pesticidal RNAi works in pests and non-target organisms, it's difficult to predict how this technology might affect the environment. Sounds dire, right? Well, it doesn't have to be. To manage these risks, we can already take some unknowns out of the equation to make sure that the pesticidal RNAi poses minimal threats to the species that we want to stick around. When you want to eliminate the unknown, what do you need? More data. You have to take steps to weed out RNAi that silences genes other than the one you mean to target, and be sure the genes they target are really involved in the cellular function of interest. So assessing an area's bioinventory can help. Then we'll know what species might be exposed to RNAi. Also, we'll get the genomes, or genetic blueprint, for the exposed species, and screen potential RNAi molecules to see whether a particular pesticide might hurt the species we want to keep healthy. From this, we can develop comprehensive risk assessment procedures that can make sure our desire to manage pests isn't coming at the expense of Mother Nature. Science can help us to understand the benefits and risks associated with this amazing new technology called RNAi, and where it fits in with a sustainable and long-term successful plan for agriculture.